Okay, hello and welcome to our molecular genetics exam review. Remember, this is going to be covering chapters 16, 17, 18, and 20. And we will be kind of going through the same type of format where I have questions for you to look over and then we go from there. So here's my first question for you. Who discovered that DNA and not protein was the genetic material? I'll give you a second to look at it. Messelson and Stahl, Chargaff, Hershey and Chase, or Watson and Crick, or my mom. Hopefully you went with Hershey and Chase. Messelson and Stahl were the two that found out that DNA was replicated in what we call a semi-conservative manner, where each new DNA strand consists of an old one paired with a newly replicated. Chargaff is a scientist who kind of realized that the number of A's were equal to the number of T's, and the number of C's were equal to the number of G's. And he realized this through measuring kind of the weight of these chemicals. So if I gave you the number of A's, you should be able to figure out the number of T's and then tell me the number of C's and G's, like if we did it in a percentage type of format. Hershey and Chase were definitely the ones that discovered that DNA and not protein was a genetic material. Remember, they used viruses that were had their protein labeled radioactively and then DNA that was labeled radioactively, and they checked the bacteria to see where this radioactive label was, and it turns out it was the DNA that was inside the bacteria and not the protein. So this is how we know that this is the genetic material. Watson and Crick discovered the structure of DNA, the double helix, and A's paired with T's, C's paired with G's, but they at that point did not. Well, they had just found out, I think, that DNA was the genetic material. All right, here's our next question. What types of bonds would you be breaking if you used restriction enzymes to cut DNA? So we talked about this a little bit yesterday. I'll give you a second to look at it. Hopefully you went with covalent. So hydrogen bonds are what connect our bases together here. So C's and G's and T's and A's and D's are hydrogen bonds. The covalent bonds are what actually connect the sugars and phosphates together and the bases to the sugars and phosphates. Ionic bonds are charged bonds, and disulfide bridges have more to do with protein folding than anything else. But a restriction enzyme would cut maybe between bases. And so you're not cutting these horizontal hydrogen bonds, or I guess in this case they are horizontally, but you're cutting the latter sides, the covalent bonds between the sugar and phosphate backbone. Okay, so here we have an image that kind of walks you through this. So what you're actually cutting are these covalent bonds, and then the hydrogen bonds get disrupted as well, but they are not physically cut. All right, next question. Why would we need to cut our DNA before running a gel electrophoresis? I'll give you a second to look at the options. So hopefully you marked D. A is sometimes true as well. Exponentially replicating DNA would be what what we talked about with PCR, or polymerase chain reaction. This is when we have a tiny bit of DNA, maybe from a fossilized specimen or something, that we need to amplify so that we can use that DNA to create additional tests. We don't need to combine it with bacteria all the time. Sometimes that's what we do. But really the reason we cut our DNA is so that we can see different segment lengths of DNA and see who has that same segment in common. Okay, so here's what a gel electrophoresis would actually look like. And remember, you would load all of your DNA into this top part, hook it into a power source, and then watch over time as these different bands spread out. Your shorter molecules should move faster than the longer ones, which should make sense. And that's a gel electrophoresis. All right, next question. Which of the following is not a characteristic of DNA structure? So it says strands are anti-parallel, 4 nitrogen bases, purines pair with purines, double helix shape, and bases are connected by hydrogen bonds. Give me a second. Hopefully you chose C here. The strands are anti-parallel, and what that means is if you have DNA that's running in the 3' to 5' direction, the other strand runs in the 5' to 3' direction. So they are running in opposite directions. This is referring to the sugar phosphate backbone orientation, and that's just what we call them. We do have 4 nitrogen bases. Hopefully this is pretty straightforward by this point. We have adenine, guanine, cytosine, and thymine. We do have a double helix or a twisted ladder shape, and the bases are connected by hydrogen bonds. The only thing that's off here is purines pairing with purines. This does not happen. Your examples of purines are A's and G's. These are usually your double ring structures, right? So they can't pair with each other or DNA would be too wide. So what a purine pairs with is a pyrimidine. And a pyrimidine would be your C's and T's, and these are your single ring structures. And so when you have a single ring that pairs with a double ring, then you get a uniform width throughout your DNA, which is why G's pair with C's and A's pair with T's. So a purine always has to pair with a pyrimidine. All right, next. How are DNA and RNA similar? Okay, so we have they are both double-stranded. They both utilize 4 nitrogen bases. They both use uracil. They are both stuck in the nucleus, or they have deoxyribose phosphate backbones. Only one here that is correct and broad enough to be correct is that they both utilize 4 nitrogen bases. If we walk through the rest of this, only DNA is double-stranded. RNA is single-stranded, right? Only RNA uses uracil in their sequences. DNA uses thymine. DNA is the only one that's stuck in the nucleus, which is why we need RNA in the first place. And DNA, the D stands for deoxyribose. The sugar that's in RNA is simply ribose. So the only one that works is B here. DNA has G's, A's, C's, and T's. RNA has G's, A's, C's, and U's. So replacing T's with U's. Okay, next question. Which enzyme adds new bases onto a growing strand in DNA replication? We have helicase, primase, ligase, DNA polymerase, or topoisomerase. I'll give you a second. Hopefully you marked DNA polymerase here. So helicase, remember, is what's responsible for unwinding the DNA. Okay? Topoisomerase is responsible for preventing overwinding so that you don't have knotted up, damaged DNA. Primase brings in the RNA primer to attach to your DNA to kind of get things moving. And ligase is what comes in at the very end and glues all your Okazaki fragments together. So the only one that works here is DNA polymerase. Now in your book there's DNA polymerase 1 and DNA polymerase 3. I'm really only concerned that you know that DNA polymerase is going to be adding new bases on. So it's responsible for that. All right, here we have an image of what this might look like. So if we look up here first, here we have DNA being unwound. So the helicase is working in here to unwind this DNA. Topoisomerase are working on either end to keep this from overwinding. And then you have your DNA being replicated. So if we kind of focus in on this half, it makes it a little easier to see, right? So here we have our helicase right here, okay? Here we have our primase. It's adding on this RNA primer to kind of get the process started. And then we have our DNA polymerase. We have it here, we have it here. Okay, and this is the main DNA polymerase we'll talk about. So if your helicase is unwinding towards the left here, we are going to be looking at the directions here. So if this is 5' at this end, we have a 3' end over here. This is 3' at this end, we have a 5' end over here. And so what we have going on here is your DNA is replicated in the 5' to 3' direction, meaning that I can only add on new bases here to the 3' end. And this is how RNA works too. You can only add on new bases to the 3' end. Okay, so I know my DNA polymerase is going to be active right in this area and then continue to move down as helicase continues to unwind. Okay, this is called my leading strand because it's kind of leading, it's following right along with the helicase. It's leading the direction. All right, now if I look at my other strand, I have 5' down here, which makes this 3'. And then on the other end, I have 3', which makes this the 5' end kind of here, here, all of these little sections. And so because DNA can only replicate in the 5' to 3' direction, it has to go this way, right? But helicase can only unwind so fast. So you can see here as helicase is unwinding this way, you get a new primer here, and DNA proceeds to the right. Now, this right here is probably the first place that the helicase started unwinding, and so this is where we have our first fragment. Here's another place where the helicase unwound, so DNA polymerase could add on here. Here's another place where the DNA is unwound. So you have all these resulting fragments called Okazaki fragments in the only direction that DNA can replicate. So what you have to do then is at the end, DNA ligase comes in and kind of connects all of those fragments to make a continuous strand of replicated DNA. All right. What is the central dogma and why do your cells follow this pathway? Well, the central dogma goes from DNA to RNA to protein. Okay? Our cells follow this pathway for a reason. So DNA to RNA is transcription, right? And if something's transcribed, remember it's kept in the same language, it's just copied down. We've already talked about the differences between DNA and RNA. So RNA is single-stranded, DNA is double-stranded. DNA is trapped in the nucleus. RNA can escape. And so DNA is transcribed into mRNA or messenger RNA so that that RNA can be responsible for getting that message out of the nucleus, right? The nucleus is the headquarters of the cell, but it's not where all the action takes place. So you got to get out of the nucleus. So that's the point of transcription. Translation is when we take RNA and turn it into a protein. Now, the protein is the language that your cells understand. It's the language of the message. But your RNA has to be translated into protein for your cells to understand that message. So this is the process of translation. I would recommend looking at what are some similarities between transcription, translation. What are some of the major differences that you might see between these two processes? Make sure you understand why transcription and translation have to occur and where they happen, because that might be something that you see again down the road. Oh, maybe on your test day. All right. Okay. Next question. Why do we read mRNA in 3 base codons? Okay, so I have some options here. Because that was Watson's favorite number, anything bigger wouldn't code for enough amino acids. Should say anything smaller wouldn't code for enough amino acids, or it's just a random number that scientists adopted and use to this day. I'll give you a second. Hopefully you came up with C. Anything smaller wouldn't code for enough amino acids. So this is where we said, okay, I have 4 nitrogen bases. I have 4 bases that I can use here. If each base coded for a single amino acid, I would only be able to create 4 amino acids, which we know is not enough for living things. We need at least 20, okay? So then we said, okay, let's say each codon consisted of 2 bases. Well, that would give me 16 different possible combinations, which once again is still not enough for our 20 amino acids. So we get to this 3 base codon, and when we look at 4 bases in combinations of 3, we actually come up with 64 different possibilities here. And so this codes for our 20 amino acids and then some. Which, shockingly enough, leads me to my next question. What do we mean when we say the genetic code is redundant but not ambiguous? Well, I just told you that we can create 64 codons, but we only need 20. So this leaves us 44 extra codons. How do we make these represent something? Well, that's why we say the genetic code is redundant. Because if you look at, a coding sheet like our coding wheel or the coding table in your textbook, you will see that there are several amino acids that are coded for by different codons. And so you could have 6 different codons, 6 different 3 base sequences that code for a single amino acid. So that's redundant, right? It's repetitive. You have more than one, in almost every case, codon that codes for the same amino acid, right? But it's not ambiguous. Ambiguous means it's unclear. And every time we see AUG, we know exactly what amino acid that codes for. It is never ambiguous. It is never unclear. We know exactly what it is. So that's what we mean when we say the genetic code is redundant but not ambiguous. All right, let's keep moving on here. What does not happen during RNA processing? I'll give you a second to look. We have exons are removed and introns are connected. Introns are removed and exons are connected. 5' cap and poly A tail are added, or the RNA strand is simplified. Which one does not happen? All right, hopefully you went with A here. The exons, as weird as it is, and I don't know, well, I do get why they named them that. The exons are the genes that you want to keep because they are the genes that are expressed. All right, so X in this case stands for expressed. I have no idea why they named the introns like they did, because the introns are the ones that you actually want to splice out, to get out of there. The introns are non-coding segments. They do play a role in like DNA fingerprinting and stuff. They do not play a role in sending messages, okay? So you remove the introns, keep the exons, which simplifies the RNA strand, and then you process that mRNA by adding a 5' cap and a poly A tail onto either end. And what this does is it protects the mRNA from all of the enzymes out in the cell that are going to immediately want to start degrading it. All right, so we're getting close here. If you were to choose a mutation to occur, which would you choose? So, in other words, which one of these mutations is less harmful? Would it be a base pair substitution, base pair insertion, base pair deletion, frame shift, a nonsense, or a missense mutation? I'll give you a second. I would probably choose a base pair substitution because just substituting an A for a G, for example, oftentimes will give you the same amino acid. It depends on where that substitution takes place because remember the genetic code is redundant. So a lot of these codons code for the same amino acid, okay? A base pair insertion or deletion is always kind of a scary idea because if you insert something, it's going to shift that whole reading frame, which is what we call a frame shift mutation or a reading frame shift mutation. And it's going to change every single codon after that point. Same happens with a base pair deletion. If you delete a base pair, it changes everything after that point. A nonsense mutation is where a message is started and then it's stopped before you get to the end. So it's an incomplete message, which is usually non-functional. A missense mutation is where you have different amino acids in certain places and you have different messages being sent. So the one that could potentially be what we call a silent mutation is the base pair substitution. All right. Which of the following would be responsible for turning on gene expression? Well, let's think about this. If your gene expression is off, it's in the off switch position, which one of these would you want to turn it on? A repressor, an operon, an inducer, a promoter, or a co-repressor? Hopefully, you went with inducer there. All right, so if you are inducing something, you're starting it, you're getting it moving, okay? A repressor is holding something back or stopping it from happening. A co-repressor is just kind of assisting with that. An operon is your actual area for gene expression. It's kind of the segment of DNA that you're looking at for sending a message. And the promoter is the region in your DNA that's kind of like the initiating region. And so this is how your RNA polymerase knows where to add on to so that it can transcribe and translate. Okay? All right. So that's the end of our slideshow. A couple other just kind of recommendations. I would look over actual images from transcription, translation. Make sure you understand what's happening there. Look over your images for DNA replication. These are pretty big ideas that are easy to get confused. I would also make sure if I ask you to start with a DNA template strand and transcribe and translate it, that you are able to do so. And you can look at our transcription translation activity. You can look at practice problems in your textbook or your study guide, but those would be my suggestions. Okay, I hope this helped, and I hope I can figure out how to post it to YouTube. And I wish you good luck on your exam.